interphase, which starts right here where my finger is, and goes all the way around to where the first yellow line is. There's three parts to that. There's the G1, S, and G2, and that's all, again, called interphase. There's one final thing about this. There are cells in your body, and I'll talk about those later on next week, and later on in the semester, that go over to here. And all they do is survive. They're weird cells, so they don't divide and reproduce. In fact, there are cells in your body that are older than you. What's your age based on is the day you were and the time you were born, right? There are cells in your body that were there before you were born. They're there with you today, and I'll give you an idea of how old they are. They're 137 years old in my body. Right? So that's how old these cells are, because I'm 136. Anyway, so the cells in and those are called neurons. Once the neuron undergoes mitosis and cytokinesis, and it grows and matures, it enters into this phase, and it never does any of this. All it's doing is basically what neurons do, and we'll talk about that later on this semester. So there's G1, S, and G2. Those are the biggest parts of the phase. When we're talking about the top part, or the cell cycle rather, when we're talking about the top part of this is called mitosis, and then this arrow there is cytokinesis, and that is important as well. And you see on the left-hand side, there's mitosis and cytokinesis, the steps in mitosis, and then cytokinesis is at the bottom. Mitosis deals with one part of the cell, cytokinesis deals with the other. So interphase basically has the cell increasing in size, replicating organelles. Give me an example of an organelle that has its own DNA so that it can replicate itself, like mitochondria. Replication of DNA. Also, where is the replication of DNA? Now, the DNA that I just mentioned is called mitochondrial DNA, and that is inherited from your mother, your maternal line, and that's nearly a complete clone of that maternal line. Replication of DNA in that third bullet there is nuclear DNA. DNA is found in your nucleus. We'll talk about what centrioles. I mentioned centrioles yesterday. They're duplicated. They're part of the cytoskeletal elements, and they are important in the process of cell division. In terms of phases or parts of interphase, there you see there's G1, S, and G2. G is gap, so there's the first gap phase, the second gap phase. Generally speaking, normal cell activities include protein synthesis, lipid synthesis, cell growth. All of those things, those types of things is what we're talking about here in G1 and G2. The S phase is when DNA is replicated, and once again, this mention of DNA and this mention of DNA is nuclear DNA. So you really want to distinguish what we're talking about there. So DNA in your nucleus is different than DNA in any organelles. So DNA in your mitochondria. And then it says there's also cells in that G0, the G0 phase or G0 phase. Okay. Cell division. Once the clock has struck the start of cell division, it jumps into this 4 steps. And as you get into cell biology, there's many types, especially for prophase. There's lots of different steps and sub-steps in prophase. I'm not going to worry about that. We're going to say there's prophase, metaphase, anaphase, and telophase. When you were kids and you were playing games, you're making an art project or something. You take all your stuff out, throw it on the table, right, and you start drawing stuff. And I know all of you guys would all very, very carefully clean it all up so that your parents didn't have to, or your guardians didn't have to clean up after you. That's not my daughter. My daughter is like, she's your age, and she's awful. Oh my God. She's like, she comes in and there's just a tornado follows her. And I was like, hey honey, can you clean this up? What? That's not mine. It's like,
How old is she?
she's a senior in high school. That's not mine. Those booty shorts are not mine. They're not mom's. I don't know. Those socks are not mine. They're not yours. They're not your brother's. He's not even here. Oh. Anyway, so she's awful, right? Now, you guys I know definitely cleaned up, except for Gianna. She didn't clean up. She's awful too. But here's what I want to draw the analogy to. Prophase is when the cell is setting up to do all the work. Metaphase and anaphase is doing the work. Telophase is cleaning up and being done. What happens in prophase? There's three things. Number one, the nucleus, which has a nuclear envelope, the nuclear envelope disappears. Number two, the DNA, which was present in the nucleus but invisible because it's very, very long and light passes through it. It's so long and stringy. The nuclear DNA condenses it. It's as if I took a shoelace and I went like this and I twisted it. If the shoelace was 72 inches and I twisted it, it could go down to 5 inches long. But because I twisted it, it's really, really thick. So the DNA then gets condensed into chromosomes. And then the last thing, and that is the centrioles. Right here, the centrioles migrate to the poles of the cell. Now, the poles of the cell are equivalent to, where does Santa Claus live?
North Pole.
Okay, so the North Pole, who lives in the South Pole? Penguins, right? So penguins, and it's as if Santa Claus is up and the penguins are at the equator and they separate, right? Okay, so in terms of, that's what's happening in prophase. In terms of what's happening, there's subparts of prophase. There's prometaphase, there's early prophase, there's late prophase. Don't worry about that. We just worry about prophase. Metaphase, chromosomes line up. If you guys are all chromosomes, and this was the center of the cell, I would line you up, pair you up, and then this is right down the middle of the cell. This is called the metaphase plate, or the equator of the cell. Third step is anaphase. The chromatids or chromosomes move to opposite poles. So now I line you up here, and then where are the poles? Well, the centrioles are over there, and you're going to go over towards the centrioles. You're going to separate. Half of you are going to go one way, half of you are going to go the other way. And then we're going to clean up the mess. Right? At this point, everyone over here is going to get grouped up into a nucleus. Everyone over there is going to get grouped up into another nucleus. That nuclear envelope is going to get reformed, and then because you're really, really tightly wound up, you're going to unwind. And that process of unwinding allows the cell to then use DNA for what is it supposed to do? For protein synthesis, for lipid synthesis, carbohydrate synthesis. So making stuff. So the unraveling of the DNA basically allows it to get used. So whatever happens in prophase happens in telophase. Right? So this is the cleanup phase, this is the setup phase. Now while telophase is happening, and wait a minute, the entire state, all four phases of mitosis only has to do with the nucleus. While mitosis is happening, and sort of the start of telophase, and certainly during telophase, the cytoplasm and all of the organelles, cytoplasmic organelles, and the phospholipid bilayer of the plasma membrane starts to separate, and that's cytokinesis. So this part right here happens somewhere around sometime during telophase, towards the end of metaphase. Okay? So now I'm going to show you some pictures, and at the top are actual photomicrographs, and at the bottom are drawings of what's happening here. If you look over here on the left, This is the nucleus, and you can't really see much in here. What's this little dot right there? That's the nucleolus. Here are other either inclusions or mitochondria. I can't really tell. But this is a good cell. With the cell membrane, there's other stuff in the cytosol. You go over to here. Now there's prophase and prometaphase. We're not worried about that, right? We're not worried distinguishing here. This is one step here. What happens here? There's three things that's happening. The nuclear envelope is disappearing. The chromosomes are winding up and turning into chromatids, and the centrioles are separating. They're moving to where? To the poles of the cell. So the centrioles are moving to the poles of the cell. So now, does it have to be in the upper right and upper left and the lower right? No, it could be over here, it could be up on top, doesn't matter. They just separate. Those centrioles then have spindle fibers. Those centrioles have cytoskeletal elements that are attached to these things right here, and then they pull them to the end, towards the poles. So basically, there's ropes on the chromosomes, and they're pulled towards the rope. That's what's happening here. Oh, sorry. That's what's happening here. So the chromosomes at this point are lined up on the metaphase plate. This thing right here is called the metaphase plate. These are the poles. Right? And then what's the next step in this whole process? That's anaphase. Where my pointer is right now is where the original metaphase plate was. The chromosomes, now these are genetically identical, right? These are copies. The chromosomes are migrating. There's three statements, at least three statements that mean the same thing, but they're saying very different things. Number one, the chromosomes are migrating away from the metaphase plate. They're migrating towards the poles. They're migrating towards the centrioles. They're migrating away from each other. All those things say the same thing. Basically, they're going this way. As they go that way, and the further they get away from that, and again, they're getting pulled there, right? Once they get sort of close to where the soon-to-be nucleus is going to be, which is going to be right about here, once they get close enough there, they start unraveling. The nuclear envelope starts to get built, and then something called a cleavage furrow. That cleavage furrow is where there's a little indentation in the original cell, this cell right here. Now this cell has 2 copies of DNA. You don't want 2 copies of your DNA. You want 1 copy in each cell. So you separate it, and as you separate it, the cells start to become distinct, and then they're genetic, now they're separate, right? This cell, if it's a good functional cell, this cell has all the copies of the DNA that this has all the organelles that this have, everything should be identical. And then whatever these 2 things they go on to do is up for debate. Okay, so those are all the steps in mitosis and cytokinesis. Cytokinesis has to do with the cytosol, the plasma membrane, and organelles. Mitosis, just nuclear DNA. If you look at the top here, these are actually pictures at the top of, I think they're whitefish cells. Generally, they're, so these are photomicrographs of actual cells. Here's the cells that are nearly complete separated, right here. Here's the cleavage furrow right there where my pointer is. This is the cells that are the chromosomes that are separating, so where my pointer would be the metaphase plate or the equator of the cell. We were talking about cells that were growing and dividing, but sometimes we have cells that don't do well. In fact, you want to have areas where there's something called apoptosis. Apoptosis is programmed cell death. And that's not individual death, it's programmed cell death. So, if I were to go back to you and take an MRI of you in utero, you would have had little limb buds, and you would have little flippers. Literally, you look like little flippers. You had gills. Oh, by the way, I had it too. You had gills. You had big eyes. You had a little head. And those flippers look like fins. They're like this. They're called limb buds. How many people have limb buds right now? Right? Hopefully, probably you have this. Right? When you had limb buds, we had one set of cells that was a single thing. Right? Now, we can separate them. We don't have any cells between here. Where the hell did the cells go? Anyone that were there died. Okay, so we developed this as a paddle, and now we separated them, and we have cells that die. So that's called apoptosis. Now, sometimes the cells are dying because I mentioned earlier on the previous slide that the cells are, if the nuclear DNA and the cells are perfectly intact, everything's great. But if the cell has a genetic damage to it, what could that potentially happen to it? What could that be called? If there's nuclear damage to the cell, to let's say that cell right there, what consequence could that have to the body? If there's nuclear damage to the DNA in the cell that I'm pointing at right there, what could that cause the body? What could happen to the body?
It could create bad mutations.
So there's damage to the damage to the DNA would be a mutation, and that mutation changes the cell, and the cell activities now are not controlled. What's that called?
Cancer.
It can be called cancer. There's other diseases as well, but one example is cancer. I talked yesterday mentioned cystic fibrosis and the production of mucus, too much mucus, and there's too thick mucus, and it prevents any normal activities. That's another example of the problems with those cells. Yeah.
What about like too much production of protein?
I don't know about too much production of proteins, but definitely wrong types of proteins or wrong structure of proteins. That will be another example of the cell not operating correctly.
How are these cells different from the cells we just studied? Like, because these cells have chromosomes and a nucleus, but they don't have the ribosomes, they don't have...
No. These cells are, so the ribosomes, the cytoskeletal elements, the mitochondria, everything's in here, right? So it's just not drawn in here, right? So you assume that these are all good cells, okay? So there's definitely time for cells to live and then other cells to die, so that there's definitely things that happen. Now, there's other examples when cells get damaged. What happens when cells get damaged? Well, if they're damaged internally, then there are specialized cells that are part of the immune system called macrophages. Now, look at that suffix there, PHAGE. What does a cell that does PHAGE do? What does that mean?
Like eat.
Not like eat. This cell eats. It eats big things. This is an example of phagocytosis. This cell undergoes phagocytosis. Its job is to engulf things. So that PHAGE means phagocytosis. Should remind you of that, okay? So there are times when cells get damaged, you need to get rid of them, or get rid of them, break them down, and then have other cells that undergo mitosis and cytokinesis and rebuild or replace new cells. If any of you have ever been cut by anything and you bled, that you hopefully aren't bleeding at the moment, right? That cut has been healed. So any of the damaged cells, macrophages ingested them and got rid of those damaged cells, and then through normal cell division, mitosis and cytokinesis, you replace those damaged cells and you repaired that tissue. This is always the worst part of these lectures. Effects on aging. I feel like I'm aging in front of you. When you talk about aging on the cells, generally speaking, cells just don't do as well. Individual cells, don't be saying that to me. Alan's going like this. I'm gonna start crying. So generally speaking, aging cells as they get older, they don't do as well as they did when they got younger. When they're younger, there's a number of different changes on it, and there's a number of reasons why, including DNA damage and mitochondrial damage. Those are two major things. And that is really how I would sort of spin a question like that. All right. Questions about chapter 3? Aside from the questions that weren't supposed to be on the quiz today, because I messed up, aside from those questions, does anyone have any issues or questions about the way I worded questions or what I said about the questions? Is there anything surprising about quiz 3? Quiz 4? One thing about quiz 4 is that we've upped the game here. So if you just go back a couple weeks here, first quiz was an open book quiz. Second quiz was just sort of list of terms, right? And it should have been pretty reasonable here. The third quiz was the chemistry quiz, and this cell quiz. So we're upping our game here. The requirements are increasing. Next time you are evaluated will be all of that stuff plus the stuff that's coming. And we're going to start that right now. Okay? Any questions? All right, let me just switch to, guess what I'm going to do? I'm going to start drawing. I'm not drawing a lot today. Sometimes I just do this on the board and I don't really do much, but today what I'm going to do is the school purchased a new iPad, so I'm going to try to use that today.
Was it Rhina or Ryan?
Right now. Wait a
I can look there, I guess.
What?
What the exam is gonna be based off of is gonna be quiz 1, 2, and 3.
So the exam will be on quizzes 2, 3, and 4. Not quiz 1, right? That's it. And then anything that we talk about for chapter 4, which is tissues. Exam, so the quizzes 2, 3, and 4, some of those same questions will be on the exam. All right? Some of those, so if you go back to these lecture notes now and see what I talked about, you should go back to those quiz questions and you should be able to answer those questions about mitosis and cytokinesis that you're going to get credit for anyway. Right? But you should be able to answer those, right? And then that's a great way to study. You're going to see those same questions on the exam.
Wait, so if it has anything to do with what we didn't learn, and we get it right, it's not gonna count? Is that what you said?
Could someone answer that question? Is it going to count? You're just making things up. It's definitely going to count.
Okay, but if you get it wrong, it's not gonna count?
No, we're gonna count it because we don't know.
All right?
But we didn't, what are you talking about? I'm so confused.
So-
No, Rich, I'm saying, right now, the quiz we just took.
Gertrude.
Oh,
Okay.
Dr. Fancy.
Oh, okay.
The quiz we just took,
Juliana. Yes.
the things that you put on the board, if we get them right, even if we didn't learn them, do they count?
Yes.
And if we got them wrong, they still count?
Yes. It's as if so everyone's going to get those corrected, either way. Okay, what else can I do? The alternative would be to just do it out of fewer questions and it'd be a real pain in the neck for me, so, no. So, all right, so let's transition here to
Dr. Fancy,
yes.
do you remember Blanca?
Do I remember what?
Blanca.
Yes. You know what I used to call her? I don't know why.
White.
Huh? I started calling her one day in lab, Bianca. And I was like, what the hell did I do that for? And she started laughing. And then it got really bad because then I was like, Blanca Bianca, and I was like, oh crap. And then I started getting confused, and anyway, it was, yeah, so, no, I don't remember her. Why? What?
I'm asking because did she do good in the beginning? Or was she just like us getting like,
All right, so now, did anyone in this room think I'm gonna answer that question?
No.
No. If I talk to her about her
Wait,
her
I
grades,
want the
yes, I
volunteer.
will.
was the last
What?
class struggling as much as we were struggling now?
So every class has struggled to this level. There's maybe one or two classes that haven't, but that is a rarity. And I've only been teaching this since 2013
Oh.
here, right? And I've taught it before that too. By the way, you know what the failure rate would be on the main campus for this class?
50%?
About 50%. So if there's 200 students in the class, about 100 of them are gonna fail.
Okay, now I don't feel too bad. Right, I don't.
If there's 200 students in the class at URI, about 100 of them will fail.
Let those college students fail.
So that, I'm not trying to, I'm not trying to scare you. That's really what the truth is. I mean, that's,
Well, then how did they pass the class if they failed? They had to retake the class?
yes. Or they change their major.
Oh.
So does anyone in this room want to have a nurse that doesn't know what the hell a tissue is or anatomical position, doesn't want to know doesn't know what the femur is or a muscle? Does anyone want to have a nurse that's like that?
No.
Yes.
100% no. Wait, what?
Do you want to have a nurse that doesn't know what the hell he or she is talking about? No. Do you want a doctor that doesn't know what the hell they're talking about? No. And I can tell you that I hope that this former student comes back to talk to you guys and to the 11th graders and younger students too. But she said to me, Dr. Clancy, I wish I could go back to school because your class was scary. But you want to know what's scary? When you're on, and she's swearing at this point. She's like 28, right? So it's not a big deal. 25, I don't know how old she is. She's a grown-ass woman. So she's like, Dr. Clancy, when you're on the floor and you go into a patient's room and there's things going off and there's stuff on the floor, and she's describing it, right? And it's like, that's gross. Anyway, she's like, and there's no book, there's no one around, there's no one on the floor, it's you and your patient here. That's scary. Once again, if,
That's unfortunate.
well, and it happens, right? Literally shit happens. If you can't handle that, if you can't handle this, you're not gonna be able to handle that. Okay? You can handle that. Now, if you're not doing well right now, things are not over, right? It's not, we're not done. You can do this.
No, y'all be fine.
And I also want to remind you that former students say to me, Dr. Clancy, you do too much for the students. Don't do so much. All right?
Who said that to you?
Who said that? A lot of students. One of them is this former student who said to me, you make it too easy for them. So when we start talking about chapter 4, which will be on Monday, we'll finish. Monday and Tuesday, we'll definitely be done with this, right? We're gonna try to finish on Tuesday. It'll be a long lecture on Monday. We'll try to finish on Tuesday and have time for a review. But we're gonna have the exam on Wednesday. There's four tissues that we're gonna talk about.
This makes me just want to give up.
I label them 1, 2, 3, and 4. I'm gonna put 2 in red and 2 in blue. The 2 in blue, if you go to the lecture notes,
You know what's crazy? You change your major. You're basically changing your whole career. No.
yes. Well, I mean, you can go back to nursing if you want, right, later on. But generally, yes, if you start studying business, then you're probably not gonna be in medicine. All right? The two in blue are two different tissues that we generally are not going to focus on a lot. And when I say a lot, I mean we're not gonna focus a lot in this chapter. We're gonna focus a lot in the coming months here. We're gonna have a whole unit on the muscular system, and I'm gonna do an entire, very, very detailed diagram on the muscular system and then the nervous system as well. Let me do this. So let's start talking about these tissues and what are some of the characteristics of each of them. So when you start talking about these tissues here, we're going to talk about the cells. We just got done talking about the cells. So there's cells and cell types. How many different types of cells and cell types do we have? We're going to do something called cell spacing. You guys are not sitting on top of one another, right? There's a little bit of space between you. That's what I'm talking about here. There's some cells that are right next to, close to another, and then others that are not. So this is a very, very brief look at some of the major characteristics of tissues. So we're gonna start doing this I'm gonna do it for epithelial tissue and connective tissue, muscular tissue and nervous tissue. When I get to the muscular and nervous tissue portion of the lecture, we're gonna go over the whole thing maybe in 4 or 5 slides, something like that. And this chapter is by far and away the longest chapter of the semester so far. So that means that the stuff that we're going to be talking about basically deals with epithelial tissue and connective tissue. The epithelial tissue has one type of cell. There's one type of cell, and in contrast, connective tissue has many, many types of cells. There's about 9 different types of cells. The epithelial tissue is tightly close packed together. They're always next to one another. So that if I pushed all of you, so there's 5, in this second row right here, there's 6 people in the row. If I pushed everyone to the right against the wall, and then I put another 5 people in this row, in this second row, right? That's how closely packed these cells are. So the cell spacing then is 0. So the cells are tightly packed. So there's no spacing between cells. Connective tissue has a lot. There's 3 shapes of epithelial cells, and there's many shapes of connective tissue cells. And we'll talk about what those 3 shapes are at the bottom of this. In terms of cell layering, for epithelial tissue, there's either simple or single, 1 layer or multiple layers. So there's either 1 layer or multiple or many. Because connective tissue is so varied, it's hard to talk about that, so we're not even going to talk about that. Where do you find epithelial tissue? You find it in one place, always, it's adjacent to what's called the lumen on the body. So the lumen is a space. So before I get to what the space is, we'll get to talking about functions here. Connective tissue, as you can guess here, connective tissue is all over the body and has a variety of areas here. Similarly, the functions of connective tissue is really, really detailed. And they go back to the 9 types of cells that I have on the left-hand side of this drawing, right here. So there's 9 types of cells, and actually we can divide it more than that. I'll do it for 1 second here. So there's many types of functions. Epithelial tissue has 2 types. Yes? Lumen, L-U-M-E-N, and I'm going to tell you what that is in 1 second. There's 2 functions for epithelial tissue. There's a number, so the first function is protection, and it can either be, and then the other function is it's called diffusion. It allows things to pass through. There's a number of different terms we can use, but for right now, I'll talk about diffusion. And then, what is a lumen? Lumen is a space. Where do you have spaces in your body?
It's like the area in between your organs, there's space in between those, or like space in between your veins and arteries.
So the space between your organs, those are cavities and those are filled with fluid, right? And we'll talk about what those are a little bit, but you're getting more along the lines. So spaces in your circulatory system, there's space in there. In your lungs, there's space in there. Anyone have breakfast? That food filled space, right? And then, so all of those things are space, right? And then finally, if I have my left hand and I'm getting close to my right arm, I'm not touching it, that's also space. So there's a free surface, and then there's any internal organs with space in there. Now, when you eat food, it is in your stomach, but it's not part of your body. Right? Why is that? Because you don't necessarily digest it. You don't necessarily incorporate it. If it passes through your gut, it will be changed, right? But the molecules will be unchanged, the chemical molecules. So there's something called cellulose that you know now is a carbohydrate. Cellulose we don't digest. So you eat it, and that's also called fiber. You eat it at a meal, and then that meal, it goes into your stomach, your intestines, and it passes out unchanged. It's still cellulose, even though it looks a little bit different. I'm not sure if you noticed that, but it definitely looks different coming in than going out. All right? All right, so the lumen is space. Spaces include the body surface, the skin, the body surface. So the lumen will be the body surface, and it also include the lungs, blood vessels, and the digestive system. There's other spaces too. Okay? So now, if you are talking about, going back to the functions here. So I talked about the lumen, and that's what a lumen is. It's space. And there's examples of 2 different types of spaces: outside of the body, inside of the body. If it's outside of the body, it's protection. If it's inside the body, it's diffusion. And then there's other stuff. What is diffusion?
go from high to low?
Possible.
Water?
So, no, if I said, what is the passive movement of water? It's not diffusion, what is it?
Osmosis.
Osmosis, right? But it's a form of diffusion, right? So thank you. Diffusion is, what you're talking about here is moving stuff from one high concentration to low concentration. That's a general term where, so that is meant to tell you you're moving stuff around the body. Does it have to only be diffusion? No, it can be other things too. But we're moving stuff around the body. Right? So this cell layering here, there's either a single cell or many cell layers. If there's many cell layers, then it's protection. If it's a single cell, then it's diffusion. Okay. Can I borrow this? Douglas is gonna punch me, and I'm gonna have this in front of me, right? This piece of paper. Is that gonna hurt? He's pretty weak, but is this gonna protect me? I don't need the protection, right? Because I'm so big compared to him. But this is not a very big source of protection, right? If I took a whole bunch of them, this is a different form. Maybe not, right? It might not be that much. It's certainly better than the first one, right? This is multiple layers of cells that are adjacent to one another, and that's equivalent to your skin or your integumentary system. Diffusion, if a single layer of cells, there's not a lot of between them, right? There's not a lot of space between them, and so as a result, things pass through them easily. Let's talk about shapes.
Why does the shape matter?
Shape matters because it helps you determine what function is.
Suture is.
So there's squamous or flat cells, there's cuboidal cells, and there's columnar cells. Tall cells are columnar, cuboidal cells are a little square, and then flat cells are at the surface of your body. Okay, so there's 3 types of cell shapes, and I said there's layering. I said there's, oopsie, cell layering, there's either one or many cell layers here. The term when you're talking about layers of cells in epithelial tissue, if it's one, it's simple. If it's many, it's stratified. So now we can then say simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar. So we have 6 different types of epithelial tissue. In principle. In practice, we don't have that many. We have about 3 that are important. Again, I'll talk about that in a little bit. Okay? Does anyone have questions about this?
No. Just make sure you post these, so I
Yes.
can like read drawings the right way.
Good. I'm glad you're going to do that.
Okay.
So let's go to muscular now. How many different types of muscles do we have? I asked some of that of you guys at the mini practical last week.
It's 3.
3 different types of muscles. What are they?
Smooth skeletal.
Smooth, cardiac, and
Skeletal.
skeletal. So there's 3 types of cells, right? And there's, I'll just put it down at the bottom, smooth, cardiac, and skeletal. I didn't give myself a lot of room. Cell spacing, the muscle cells are tightly joined together, and so there's not a lot of spacing. There's very little spacing. The cell shapes, they can be long or they can be very long and cylindrical, or they can be sort of pancake shaped. We really don't talk about cell layering in muscles.
Pancake?
Yeah, they're pancake shaped. And there's 3 locations. You just told me where they are, right? There's the heart, smooth muscles. In one example is the digestive system, for example, and then skeletal muscle. What's the functional muscle? The only thing muscles do is contract. If it extends, they have other characteristics. But if a muscle extends or gets longer, that's not because that muscle cell is doing that, it's something else is acting on it. So the only thing it does is to contract. Whoops. Trying to move this up. All right. So, in terms of nervous tissue, we have 2 types of cells that are nervous tissue. Does anyone know what they are? What type of cell is found in nervous tissue?
Is it neuron?
Neurons, and what else?
Neurons.
Isn't it like ganglions?
So that's a part of the nervous system,
Oh.
but there's cells that help neurons, and there's 2 names for them. There's, generally I use the word neuroglia, or there's neuroglia or glial cells. The cell shapes here, we don't really talk about cell spacing here. The cell shapes there, they're either a classic cell shaped, in other words, they look like a real, like a cell that I just drew, or they have many, many, many cell extensions. Which cell, neurons or neuroglia, have many, many, many cell extensions? Neuron or neuroglia?
Wait, you said which cell has many extensions?
Yes.
I'm gonna say
Neurons?
neuroglia.
So some neuroglia do, but others don't. But all neurons have lots of
Because
extensions.
they're sending so much to like each part of the body, correct?
Well, more than that, they also they have these appendages of the cell appendages that reach out to try to communicate with other things.
Yeah. But
So you guys know what hair extensions are, right?
Yeah.
I'm gonna get some next week. Hair, so cellular extensions here, all we're talking about here, this is part of the body, right? We're extending, cellular extensions are, imagine an original cell here, and now we have these long appendages off
I
that.
have a question. Is our like head hair like cellular extensions, but technically like kind of like outside of the body? Would that be like, well, could you consider it like a cellular extension
No, it's a cellular product.
product?
Yeah, so it's generally gonna be keratin, it's generally gonna be a protein that's produced, and so it's not a cell extensions.
for shape?
Yeah, that's for shape. We don't really talk about cell layering. Where did you find neurons and neuroglia? There's two major areas.
Brain and spinal cord.
Brain and spinal cord, and then also in spinal nerves. So location here includes the brain, spinal cord, whoops, and then spinal nerves. And then, what is the don't worry about neuroglia. Neuroglia, we're going to say generally that neuroglia function to support neurons. What do neurons do?
Transmit electrical signals.
Only thing they do, just like muscles contract, neurons send electrical signals, and that's all they do.
And you said we don't worry about the layering.
Right. So that's for neurons here, and then there's support neurons. So the stuff in red is what we're going to focus on in the coming lectures. The stuff in blue, we're not. The stuff in blue that I just talked about is pretty much, I'm going to show you a couple of pictures, and we'll talk a little bit about it, and it'll be done in about 10 minutes. In terms of the questions for the exam coming up, there's going to be a total of 30 questions on each of these 4 tissues for the exam. 25 of them or more will be on the epithelial and connective tissue. 2 or 3 will come from muscle and nervous, something like that. Okay?
What is epithelial function?
Excuse me?
Epithelial function is protection and diffusion.
Yes. All right. So this is sort of a summary of this. I'm actually on URI Secure. I'm not going to save it right now and upload it, but I will upload it before I leave this room today. So now let's start talking about chapter 4. Okay? Yes. What? Is that good? Got it? Anyone have questions? So what I want to do now is I want to get into chapter 4 a little bit so that we have less to do on Monday. Okay? And then Tuesday. Okay? All right.
Don't we have like Monday and Tuesday to do this though?
Yes. But I'm concerned because we have a lot to talk about on Monday and Tuesday. So the more that we talk about today, the less we have to talk about next week. All right. So let me get out of here.
But wait, because I don't want to miss it. So then imagine in your video it catches what I said right now that I gotta go pee.
Wait, how long do we have? till?
I don't know. He said that we're gonna go over a little bit of chapter 4 and then so I think until 11.
Oh, we don't have a break?
No, I don't think so.
Oh,
Well, we need new notebook by next week. I'm ready to do the second portion of the Godberries. Well, it's the first page. Damn, this week's combo. The warmer system.
Okay, so you can anticipate what we're gonna talk about next. We're gonna talk about tissues, and we're gonna start talking about the 4 different types of tissues. What I wanna do today is really just introduce this whole concept, and then we'll jump into it really detailed next week. So when you're talking about the body itself, if you look at this diagram on the right side, all of these areas that I'm pointing that are internal parts of the body, they have spaces. And those spaces, I'm going to tell you, they have a particular type of epithelial tissue. And then superficial to that, like if you look at the stomach, adjacent to the space is called the lumen. That's a particular type of epithelial tissue. A little bit deeper from that, there's going to be a particular type of muscle tissue. And then deeper, superficial from that is going to be a particular type of connective tissue. So these tissues work together. And then, by the way, there's also the nervous tissue that also will integrate into the stomach and then elsewhere as well. These 4 tissues come together. They produce something that does one job and it does very well. So for instance, the stomach or the lungs, the heart or the blood vessels, those things do one thing very well. All 4 of those tissues operate together when they function normally. I think these numbers are correct here. So if you look at this, there's about 70 or so slides in this lecture series. There's about 25 or so epithelial, there's about 25 or so, maybe a little bit more connective tissue. The remaining are muscle and nervous tissue, and then a little bit of membrane repair. And once again, I hate this topic here. Getting old, what do your tissues do? Getting old, they start to break down. That's what's happening.
That's tough. Oh, man.
So let's talk about some terminology here. You make me cry. Yeah, talk about some terminology here. When you're talking about structure of the cells, you should have that picture of a cell that I drew on the board in memory, right? So that's the one thing you should think about. Non-cellular extracellular matrix. What the heck does that mean? I'll give you an example here. I don't have much of it. Non-cellular extracellular matrix here. So there's stuff that's outside of the cells that's made by the cells that is either in the lumen or elsewhere or internally. So hair would be one example of non-cellular extracellular matrix. Extracellular, what does extracellular mean? Outside of the cells. Cell functions, we'll talk about the cell functions, right? And I'll give you a word of warning. When we get to connective tissue, there's going to be 9. Some books will get up to like 12 or 14 different types of cells. It's connective tissue cells. I think we go to 9. I try to mush a bunch together. The vocabulary gets pretty significant there. When you're talking about tissues themselves, I'm going to show you pictures of tissues. When we talk about the pictures, I'm going to say there's a histological section or a histology. It's a microscopic study of tissues with groups of cells right here, and you have to be comfortable recognizing that. If you have a problem with a particular group of cells and you need it looked at, examined by a medical professional, that could be cause of a biopsy, and then ultimately autopsy is the cause of death. So we talked about, I mentioned epithelial tissue. Now this vocabulary, we're going to jump right into the vocabulary. There are already things that I haven't mentioned here on my previous slide. Right? So the characteristics of epithelial tissue, very little extracellular matrix. What did I say in my list here? Very little extracellular space. Extracellular is outside. That means, conversely, the cells are tightly packed. If I pushed, again, in this room, if this aisle right there, I pushed everyone in and I got another 8 people right there, they would be all smushed together. That's what epithelial tissue is. They're all smushed together, regardless of where they are in the body. They cover surfaces, they line spaces. The space, the area right here, is not, the term is not listed, but it's called lumen, L-U-M-E-N. That's the space. If it's outlining the surface of the body, the space is on the surface of the body. If it's lining the digestive, respiratory, urogenital tracts, those spaces there have fluid or solids. depending on where we are. Blood vessels and some body cavities here, but particularly blood vessels, right? What do they have? They have blood. When you're talking about the cells themselves, you have not all parts of the cells. Remember, I drew that sort of the cell, that blob of a cell, and I didn't say this is top, this is bottom, this is left, this is right. Right? But now when we're talking about epithelial tissue, we can say that. Because when the portion of the cell that is next to the space, it has one thing that's called the apical surface, and then there's the surface of the cell that's basal, is the basal surface, of the basement. I'm adjacent to the door right now, so I'm one group of cells that would be adjacent to, I'm next to the space, I'm leaving. I'm fine. I'm laughing. Where'd it go? Anyway, so I'm adjacent to the door. Right? That's what it means, apical or free surface. Some of you are adjacent to the wall. That would be the basal surface. Okay? And then finally, there's this connective tissue here that's always there that basically bonds its glue. A basement membrane is the glue that keeps the cells from being pulled off of the tissue and the underlying tissue. So that epithelial tissue have strong intercellular connections. So those strong cellular connections, intercellular connections, and intercellular connections is between cellular connections. These specialized cell connections there, depending on where they are in the body, where the cells are in the body, you can say there's going to be one type of connection or another type of connection because you want this function or that. And that leads me to the function itself. And we talked about the function, protection or diffusion. Look at these two things, these two, 5 and 6 here, characteristics. So it's avascular and innervated. Avascular. Something is avascular. What does that mean? Any of you guys ever had a paper cut? My sister-in-law owns a salon, a hair salon. Obviously, I don't have to go there very often. But she complains a lot about, not a lot, but I make fun of her all the time because she's like, oh, I got a hair splinter and it hurts. Paper cuts, do they bleed a lot?
No.
Do they hurt?
Yes.
Why don't they bleed a lot? What's that term?
Avascular?
What's the prefix A mean in front of anything?
Absent.
Absent. It doesn't, there's nothing there. No blood vessels. Why doesn't, if you just damage epithelial tissue, why doesn't it bleed? Because it's avascular. If you damage that tissue, just epithelial tissue, you feel it, right? It hurts. That's innervated, right? How does epithelial tissue survive? How does it undergo mitosis and cytokinesis? How does it undergo cell metabolism? How does any of this happen? Right? Diffusion. Material from below or adjacent to it, they get glucose from right near it and sends it to those cells that need it. How does that happen? By diffusion or facilitated diffusion. It might happen by active transport, but it has to happen. If it doesn't happen, then the cells die. When you get damage, let's say you have a cut. I have lots of places where I have cuts on my arms and legs. What does that mean? High regeneration capacity. Do I have any gaping wounds right now? When I was about 5 years old, I was at the playground and I smashed my head. I had 5 stitches right there. I don't think that's bleeding right now, right? I wouldn't be here if it was still bleeding. What happened, right? Well, first of all, the doctor kept it from, he gave me 5 stitches. But then it repaired itself. How did it repair itself and what's that called? Regeneration. So that term regenerate, in other words, means mitosis and cytokinesis. Some cells have high regeneration capacity, some tissues, others do not. Which tissue does not have high regeneration capacity? Out of the four, there's epithelial, connective, muscle, and nervous.
I think it's nervous.
Nervous, right? Can you give me evidence of that? People that get damaged,
Paralyzed and they never.
right? So if the spinal cord is paralyzed, they might be paraplegic or quadriplegic. Right? There's evidence that the damage is permanent or semi-permanent, right? Whatever. There's evidence, right? So that is an example of a lack of regeneration ability. So epithelial tissue, regardless of where it is able to regenerate, and that regeneration ability is really, really valuable. I mentioned earlier that the basement or the bottom of the cell membrane or the epithelial tissue is connected to other tissues, and without that connection, you can pull apart and separate, and that's a problem. Right? So these specialized extracellular material, what is, again, specialized extracellular material? What the hell am I talking about here? Specialized extracellular material. These things are non-cellular products that cause the cells on the basal membrane to connect to deeper structures. So what are some of the examples that I might use? So if I were to say collagen and glycoproteins, I'm not going to talk about what they are specifically, but if in a question I say collagen and glycoproteins are used by the basement membrane, are produced to form a basement membrane, what tissue are we talking about? Right? The epithelial tissue. The next question could be, what tissue would be most likely going to be connected to? And that, I haven't told you that, but the tissue that's deep is going to be connective tissue. All right? So if you look at just some of these other words right here, if you read the question here, I'm not going to ask about the structure of proteoglycans and glycoproteins. I've talked about collagen already. And I'll talk about collagen again. But I might ask a question like that. So the basement membrane also supports and guides cell migration. What is cell migration? If I have a cut and my skin is separated, right? You need to start repairing that. You need to staunch the blood flow, and without medical intervention, right? Then a scab would form. That staunches the blood flow. And then you have other cells that migrate there that start enhancing microsis and cytokinesis and enhancing tissue repair. You need guides to do that. And so that basement membrane helps tell the cells where to go. Don't go over there, go over here.
Let's say you go through surgery and stuff, and you end up getting scar tissue instead of scab. Would that be the same process?
So a scab is going to be before a scar tissue, right? So scabs will generally form as a result of coagulated blood.
But let's say like you have it covered, you have the wound covered, because you just went through surgery, so you have a patch over it, so you have it covered. How is scab gonna form if there's not like a lot of oxygen?
So if the scab is informed and your body is still able to heal, that means just there's less blood loss and there's less plasma loss. There's fewer white blood cells that are migrating there.
Would the scar tissue form like faster or slower because there's scab under it?
So generally, it's going to form faster. Yeah. Now, having said that, we're getting much more detailed in when you're talking about scar tissue in the skin, because what we're talking about here is muscle and nervous tissue, connective tissue, and then also epithelial tissue. So for right now, let's just focus on epithelial tissue, okay? Now, if you look at these two pictures right here, they're just epithelial tissue. There is at the deep area right there, there's a basal membrane right here, or a basement membrane. There's a basement membrane right here, and a basement membrane right here. Now, if I were to show you this picture right here, and I would, at the top, so what type of cell is this, or what type of tissue is it? It's not muscle, nervous, or connective. The cells are really tightly packed together. This cell where my pointer is flat. So if the cell is flat, it's a squamous. How many cell layers do we have? There's just one cell layer right here, so that's simple squamous epithelium. Is this simple squamous epithelium gonna have lots and lots of protective ability? In other words, is it gonna be able to prevent things from going across it? No. So what's its function going to be? It's probably going to be allow for the passage of certain things. This basal surface is going to be the the deep area right here is called the basal surface. This is where my pointer is the apical surface, and then this area right here where the pointer is now, that's the lumen of this tissue. So the lumen is always adjacent to the surface. So if this, or the free space, if this were, this part right here, we're in the lungs, there would be air exchange or oxygen exchange across these cells, and then getting into the blood vessels. At the bottom of this, if you look, there's many, many layers here. So this is called stratified layers. But if you look at the bottom right here, the bottom is the basal surface. And if you look at the shape of these cells, you could very reasonably come to the conclusion these are cubes or look like dice, right? And if you said these are cuboidal, you would be wrong, because you never, never, never look at the basal surface for the shape of the cell. If you look at the surface of the cell, right here, this is the free surface or the apical layer. The surface of the cell or the apical layer is flat and it's pancake shaped. Because it's flat or pancake shaped, this is called stratified squamous epithelial. So this is epithelial, this is epithelial, this is stratified, this is simple. And if you look at all the different terms on the right-hand side, I've just mentioned a lot. There's the lumen and apical surface, there's the basal surface and the membrane, there's the cell shape and the cell layering here. I haven't talked about these things right here. Intercellular space. Intercellular space is lacking. What does that mean? They're really close together. They're really close together. Because they're really close together, you also have to have something called cell junctions. What's a cell junction? We'll talk about the specifics of it. Basically, it's connections between cells. There's no discussion of, there's no blood vessels here. There are nerves, specialized epithelial tissue that synapse with nerves, and so that's why it's called innervated. We talked about regeneration already, cellular reproduction. Now, if you talk about regeneration or mitosis and cytokinesis, if you remove these in the top picture right here, if you remove for whatever reason these top several cells or these cells in the center, cells adjacent to it would undergo mitosis and cytokinesis and fill that hole, provided that they're able to, right? In other words, if you have a stomach ulcer and that ulcer is eating a hole in this and that ulcer continues, the cells aren't going to be able to be replaced. But remove that acid out of the equation here, the cells could slowly, through mitosis and cytokinesis, repair that gap. In stratified squamous epithelium, that is not the case. As you go from the basement membrane deep to this, and you go superficially, the cells begin to change, the chemical nature of them begin to change, and then ultimately, this picture is actually wrong, because these are nuclei right here, and the nuclei right here. By the time they're right here, these cells no longer have nuclei, they no longer have anything. The only thing they have is keratin. They don't have big protein here. That protein is a waterproofing protein if this is the skin, and that waterproofing protein basically causes the cell to die. So the cells up here are dead, cells down here are alive. So how much mitosis and cytokinesis can these cells go? None. Right? So the cells that are adjacent to the basal surface will undergo mitosis and cytokinesis. So regeneration here, not all cells regenerate in stratified squamous epithelium. All in simple squamous epithelium, all the cells in simple epithelial tissue will have the ability to regenerate. And this is a more complete picture and a more accurate picture of these tissues. This is actually an entire these are all 4 different types of tissues that we have here. So at the surface here, we have 4 epithelial tissues, 4 cells rather. These cells would be simple cuboidal epithelial. They're cube-like. They're not very, very, very tall. They're not long and they're not flat, right? So it's probably simple cuboidal. They have a basement layer there adjacent to the basement membrane, that basement membrane I talked about a minute ago. There's a surface right there, or a free surface, that apical layer, and then the lumen. And now let's talk about what's deep to it. Always, always, always deep to the epithelial tissue is going to be a series of other 3 tissues. So this space right here and this space right there is blood vessels. Those blood vessels are going to have some muscular tissue with it, and they're also going to have other epithelial tissue, so that's one thing. We have this major area right here, this space, where there's lots of intercellular space. There's not a lot of cells that are there. That's connective tissue. That's the second, or the third rather, tissue here. And then, not shown in this diagram very clearly, there's also nervous tissue in there. So this whole picture, although it says structure of epithelial tissue, it has all 4 tissues in here. The epithelial tissue that I want you to focus on are these 4 cells there. Let me see something here. Okay. So what I want to do now is just talk about the functions of epithelial tissue, and then we're going to stop. There are 2 functions, right? I've already told you what they are. They're either protection or passage of stuff. If it's passage of stuff, it's going to be simple. And when I say simple, it'll be simple epithelial. Then simple cuboidal, simple columnar, simple squamous. Depending on where you are in the body, if you're in the stomach, you're going to have simple columnar. If you're in the lungs, it'll be simple squamous. Right? And so that they're different here. So if you're in the kidneys, if you're secreting something, what does secreting mean? That's secreting, that's another way of saying exocytosis. What is exocytosis? You make something in the cell, you package it up through the Golgi apparatus, and then you get rid of it. And where do you get rid of it? You get rid of it into the lumen of the body. For instance, the lumen of the body, the stomach, you eject it into the lumen of the body, and then that hydrochloric acid or other things encounters the food and helps break that down. If you're absorbing it, right, all that is the opposite direction. You still have to get from wherever, from the space, you get into the cells, and then it goes there. So for instance, oxygen is in your lungs, oxygen's in the space. Oxygen has to get across that cell membrane into the cell, it has to be absorbed, and then it has to go elsewhere in the body. Here's another example here in the intestines of the small intestine of the body. So when you're allowing things to pass through the from one area to another, that's always, always, always going to be simple epithelial tissue, depending on what you have, squamous, cuboidal, or columnar. If you're going to be protecting it, right, there's a few areas of the body where you have protection. So the most obvious one is the epithelial lining of the skin. There's also areas of the mouth, the anus, right? Those things are areas for protection as well. But they're going to provide many, many layers, and they're trying to prevent the body or material from crossing that barrier. If they prevent that, then you don't have a problem. If they don't prevent it, what happens? You have an infection, or you might have an infection. Okay. All right. So we're going to stop right here. Anyone have questions about what we're doing today, what you have in store for this class for next week, Monday and Tuesday? Right. Monday is going to be a long lecture. Tuesday will be a shorter one. We'll be prepared for Wednesday. I can't stress enough, you need to anticipate what we're talking about in this class for next week because it's going to go very fast. Exam on Wednesday. Meanwhile, you have a quiz, a mini practical tomorrow, right? What's that mini practical on? Or Friday? The axial skeleton. Okay? All right, so I will see you. Now, once again, tomorrow morning's lab starts at 8 o'clock. Tomorrow afternoon's lab starts at noon. Friday mornings, they're lucky they start at 8:30. I will see you guys tomorrow or next week. Or Thursday, Friday. I'm very happy.